Hi class, this is your instructor, Skylar Huff. We are now in Chapter 20. This is Genes Within Populations. This chapter is a long one. Individuals in most populations vary greatly in phenotype, as the pigeons shown right before yourselves illustrate. These differences are often a result of genetic differences among individuals. Often the particular characteristics of an individual have an important bearing on its survival, on its chances to reproduce, and on the success of its offspring. Evolution is driven by such factors as the frequency of different alleles rises and falls in populations. These deceptively simple matters lie at the core of evolutionary biology, which is the topic of this chapter, including those that come hereafter that will be on your next test. The term genetic variation refers to the different alleles of genes found within individuals of a population. Natural populations contain a wealth of such variation. In this chapter, we will explore genetic variation in natural populations and consider the evolutionary forces that cause allele frequencies in natural populations to change. The word evolution is widely used in the natural and social sciences. It refers to how an entity, be it a social system, a gas, or a planet, changes through time. Although development of this modern concept of evolution in biology can be traced to Darwin's landmark work on the origin of species, the first 5 editions of his book never actually used the term. Rather, Darwin used the phrase descent with modification. Although many more complicated definitions have been proposed, Darwin's words probably best capture the essence of biological evolution. Through time, species accumulate differences. As a result, descendants differ from their ancestors. In this way, new species arise from existing ones. You've already learned about the development of Darwin's ideas a bit throughout the course. Darwin was not the first to propose a theory of evolution. Rather, he followed a long line of early philosophers and naturalists who deduced that many kinds of organisms around us were produced by a process of evolution. And you can see a little bit about Darwin in Chapter 1 if you'd like to read more. Unlike his predecessors, however, Darwin proposed natural selection as a mechanism of evolution. Natural selection produces evolutionary change when some individuals in a population possess certain inherited characteristics and then produce more surviving offspring than individuals lacking these characteristics. As a result, the population gradually comes to include more and more individuals with more advantageous characteristics. In this way, the population evolves to be and becomes better adapted to its local circumstances. One way to monitor how populations change through time is to look at changes in the frequencies of alleles of a gene from one generation to the next. Natural selection, by favoring individuals with certain alleles, can lead to change in such allele frequencies, but not only the process. But it is not the only process that can do so. I'll again state that natural selection, by favoring individuals with certain alleles, can lead to change in allele frequencies, but it is not the only process that can do so. Allele frequencies can also change when mutations occur repeatedly, changing one allele to another, and when migrants bring alleles into a population. In addition, frequencies of alleles can change randomly as a result of chance events, particularly when populations are small. Often, natural selection overwhelms the effects of these other processes, but as you will see, this is not always the case. Evolution can result from any process that causes a change in the genetic composition of a population. We cannot talk about evolution, therefore, without also considering population genetics, which is the study of genetic variation within population. Genetic variation is required for evolution to occur. Consequently, biologists have always wanted to know how much genetic variation exists in natural populations. The ability to ask this question has been limited by techniques available to analyze variation at different levels: proteins, genes, and now genomes. The story of genetic variation in populations is told using increasingly sophisticated tools for detecting differences. Initial approaches examine the most obvious differences, morphological variation, but in many cases, such phenotypic variation is a result of underlying genetic differences. In such cases, natural selection usually shows substantial genetic variation, such as you will see in figure 20.1, shown here now. So you're seeing a natural population of lupines from the genus Lupinus. They exhibit considerable variation in flower color. Individual differences are inherited and passed on to offspring. Other examples closer to home consider all the variation in a human population: skin, eye and hair color, hair type, height, eye and nose shape, blood type, navel. Is it an innie or is it an outie? Please don't answer that. And many more. Most human variation is a result of underlying genetic differences, as you see here. The first approach to directly assay genetic variation within populations was the use of electrophoresis to separate proteins produced by alternative alleles of the enzyme encoding genes. As each new DNA analysis tool was developed in the laboratory, it was quickly adapted for use with samples collected from natural populations. This led to, in order of increasing detail, examining restriction fragment length polymorphism, sequencing specific genes, and most recently, sequencing entire genomes. One of the most useful tools, both for genetic mapping and for the analysis of population variation, has been single nucleotide polymorphisms. These are defined as a single base difference between individuals that exist in populations that are at more than 1%. An initial large-scale international effort identified several million single nucleotide polymorphisms in the human genome, a number we now know to be a vast underestimate. Such variation is also being assayed in many other species of interest. The results for most species are similar to those in humans. The closer you look, the more variation you see. One reason for the appeal of single nucleotide polymorphisms is that we have been able to automate the analysis of multiple samples. These SNP data are now available for thousands of species, revealing the near ubiquity of genetic variation and providing tools for assessing patterns in human and natural populations. As genome sequencing moves into a phase where multiple genomes are being sequenced for many different species, a landscape of massive genetic variation is being revealed. Not surprisingly, the species with most genomes sequenced is our own. By the time you read this, more than 100,000 human genomes will have been partially or entirely sequenced. As you saw back in a prior chapter, this revealed an enormous amount of genetic variation. Individuals will have around 3% of their genome varying from the reference genome, as well as extensive variation within human populations. These studies are also documenting substantial variation between populations. This kind of work is in progress, looking at genomes from specifically Asian and African populations, and in one case, a Dutch population. The early results from these forays into human genetic variation are significant and surprising. Not only do these populations have additional genetic variation not previously seen, but they also have entire genomic regions that are not represented in the reference genome, which comes from individuals in North America. We have long known that African populations have greater genetic diversity than European and Asian populations. This is a result of human history. Our species originated in Africa. I repeat, this is a result of human history. Our species originated in Africa, and the populations there represent the oldest human genomes. A recent study that involved sequencing over 900 African genomes from different regions of Africa showed that taking all of these genomes into account, there was more than 300 millibases of DNA not found in the reference genome. It is becoming ever clearer that within a species, different populations have unique evolutionary trajectories, and that substantial variation within a species is distributed across the species' geographic range. Studies of genomic variation in natural populations are underway as well. More than 1,000 genomes in the laboratory fruit fly, Drosophila melanogaster, have been completed. Comparisons of these genomes revealed extensive variation, although interestingly, some regions of the genome were more variable than others. Just as with humans, patterns of variation differed between the populations. In another study, 133 rhesus macaques, genomes were sequenced, revealing substantial variation among these monkeys. Undoubtedly, the number of species for which individuals are sequenced will skyrocket within the next few years, substantially enhancing our understanding of population variation. Genetic variation within natural populations was a puzzle to Darwin and his counterparts in the mid-1800s. Although Mendel performed his experiments during this same time period, his work was largely unknown. Selection scientists should always favor an optimal form, and so tend to eliminate variation. Moreover, the theory of blending inheritance, in which offspring were expected to be phenotypically intermediate relative to their parents, was widely accepted. If blending inheritance was correct, then the effect of any new genetic variant would quickly be diluted to the point of disappearance in subsequent generations. For example, if a mutation made an individual 4 inches taller, its offspring, the result of the mating with an individual without the mutation, would be only 2 inches taller. And the next generation of offspring will only be about 1 inch taller, and so on until the effect completely disappeared. Following the rediscovery of Mendel's research, 2 people in 1908 solved the puzzle of why genetic variation persists. Godfrey H. Hardy, an English mathematician, and Wilhelm Weinberg, a German physician. These workers were initially confused about why, after so many generations, a population didn't come to be composed solely of individuals with the dominant phenotype. The conclusion they independently came to was that the original proportions of the genotypes in a population will remain constant from generation to generation as long as the following assumptions are met. You see those before you. There are 5 of these. No mutation takes place. No genes are transferred to or from other sources, i.e., no immigration or immigration takes place. Mating is random. Individuals do not choose mates based on phenotype or genotype. 4. The population size is infinitely large or very large. And last, no selection favoring one genotype over another occurs. If these assumptions are met, the genotype's proportions will not change and the population is said to be in Hardy-Weinberg equilibrium. You have before yourselves 2 equations. Learn them both. P plus Q equals 1. That is for allele frequency. That is how you calculate allele frequency. P represents the dominant allele. Q represents the recessive allele. P squared plus 2pq equals 1. P squared represents the homozygous dominant genotype. 2pq represents the heterozygous genotype. And Q squared represents the homozygous recessive genotype. In algebraic terms, the Hardy-Weinberg principle is written as an equation as I just mentioned. Consider a population of 100 cats. 84 of those are black, 16 are white. Black is dominant to the recessive phenotype white. The frequencies of the 2 phenotypes would be 84% black and 16% white. Based on these phenotypic frequencies, can we deduce the underlying frequency of genotypes? We surely can. As you work this out, class, you should always find Q first. As you work through this, you should always find Q first. So, first things first, what you should do is take out your calculator that you may or may not have. Press pause if you don't have it. Get it out. So what you all should do is take the square root of 0.16. That will give you the frequency of the recessive allele. Next, what you should do is take 1. Or before you take 1, we'll go to the other equation. The other equation I refer to here is P plus Q equals 1. So now that we know Q, and I say again, going backwards for a moment, we have Q squared. Q squared is 0.16. You take the square root of Q squared to get Q. So we now know that Q is 0.4. We'll now use the equation here at top to find P. P is P plus Q equals 1. So now with a bit of algebra, we subtract Q from both sides. So it would be P plus 0.4 minus equals 1. And then when you subtract Q from both sides, it would be P equals 1 minus 0.4. So P equals 0.6. So now P squared is 0.36. 2 times P times Q is 0.48. If you're not writing this out using paper, you very well may be lost. You can see this is in your textbook, and I just worked out an example for you. You will have problems like this on your test. And I did say problems, plural. Nonetheless, if we assume that the white cats are homozygous recessive, which I told you, class, we can designate the recessive B, and the black cats are either homozygous dominant or heterozygous. So I will not read the rest of this or go through the rest of this because of time. However, what I will say is you have all you need to answer this. The Hardy-Weinberg equation is another way of expressing the Punnett square described in chapter 12 a long time ago, with 2 alleles assigned frequencies P and Q. So in the figure, it allows you to trace genetic reassortment during sexual reproduction and see how it affects the frequencies of those 2 alleles during the next generation. So in constructing this diagram, we have assumed that the union of sperm and eggs in the case of cats is random. The alleles are therefore represented in the next generation in proportion to the original occurrence. Each individual egg or sperm in each generation has a 60% chance of receiving a dominant allele and a 40% chance of receiving a recessive allele. The next generation, therefore, the chance of combining 2 dominant alleles is 36%. And approximately 36% of the individuals in the population will continue to have the homozygous dominant genotype. The frequency of the homozygous recessive individuals is 40%. Well, excuse me, it is 16%, 0.4 times 0.4, or 0.4 squared, and it will continue to be about 16%. And the frequency of heterozygous individuals is, on average, 48%. Phenotypically, if the population size remained at about 100 cats, we will still see approximately 84 black cats and 16 white individuals. Allele, genotype, and phenotype frequencies have remained unchanged from one generation to the next, despite the reshuffling of genes that occurs during meiosis and sexual reproduction. One important point to remember is that the terms dominant and recessive refer only to the phenotype of the heterozygote and in no way imply that one allele is selectively superior to another. For this reason, in Hardy-Weinberg equilibrium, when selection is not operating, allele frequencies are not expected to change, regardless of whether one trait is dominant over another. So if all five assumptions for Hardy-Weinberg equilibrium are true, allele frequencies should not change from generation to generation. So in reality, most populations will not meet all 5 assumptions. So primary use of the equation is used to determine whether evolutionary processes are operating in the population and hypothesize what the processes are. So what makes populations vary from Hardy-Weinberg equilibrium? Well, natural selection. It might favor the homozygotes over the heterozygotes. Individuals may choose to mate with genetically similar individuals. There might also be an influx of individuals from other populations and mutations occurring. These things affect allele frequencies. So the way in which they affect them is shown in figure 20.4. And let's get on in that. So mutation, the 5 agents of evolutionary change is where we are. Mutation from one allele to another can obviously change proportions of particular alleles in a population. Mutation rates are generally so low that they have little effect on Hardy-Weinberg proportions of common alleles. A typical gene mutates about once per 100,000 cell divisions. Because this rate is so low, other evolutionary processes are usually more important in determining how allele frequencies change. Nonetheless, mutation is the dominant source of genetic variation and thus makes evolution possible. Look at figure 20.4a. It is important to remember, however, that the likelihood of a particular mutation will occur is not affected by natural selection. That is, particular mutations do not occur more frequently in situations in which they would be favored by natural selection. Gene flow is movement of alleles from one population to another. It can be a powerful agent of change. Sometimes gene flow is obvious, as when an animal physically moves from one place to another. If the characteristic of the newly arrived individual differ from those of the animals already there, and if the newcomer is adapted well enough to the new area to survive and mate successfully, the genetic composition of the receiving population may be altered. Other important kinds of gene flow are not as obvious. These subtler movements include the drifting of gametes or the immature stages of plants or marine animals from one place to another. Pollen, the male gamete of flowering plants, is often carried great distances by insects and other animals that visit flowers. Look at figure 20.4b. Seeds may also be blown or carried by animals to new populations far from their place of origin. Consider two populations initially different in allele frequencies. In population 1, P is 0.2 and Q is 0.8. In population 2, P is 0.8 and Q is 0.2. Gene flow brings alleles into a population in proportion to their frequency in the source population. In this case, it would disproportionately bring alleles that are rare into the recipient population because those alleles are common in the source. Thus, allele frequencies would change from generation to generation, and the populations would not be in Hardy-Weinberg equilibrium. Only when allele frequencies reach 0.5 for both alleles in both populations will equilibrium be attained. This example also indicates that gene flow tends to homogenize allele frequencies among populations. Individuals with certain genotypes sometimes mate with one another more commonly than one would expect on a random basis, a phenomenon known as non-random mating. Please see figure 20.4c. Associative mating, in which phenotypically similar individuals mate, is a type of non-random mating that causes the frequencies of particular genotypes to differ greatly from those predicted by Hardy-Weinberg principle. Assortative mating does not change allele frequencies in the individual alleles because it does not change the reproductive success of individuals, but rather changes with whom they mate. Because phenotypically similar individuals are likely to be genetically similar and thus are also more likely to produce offspring with two copies of the same allele, assortative mating will increase the proportion of homozygotes in the next generation. Inbreeding results when closely related individuals mate with each other. Because relatives are genetically similar, inbreeding will have the same result as assortative mating and increase the proportion of homozygotes. In fact, because relatives tend to be phenotypically similar, assortative mating can lead to inbreeding. In small populations, frequencies of particular alleles may change drastically by chance alone. Such changes in allele frequencies occur randomly, as if the frequencies were drifting from their values. These changes are thus known as genetic drift. Please see figure 20.4d. 20.4d. For this reason, a population must be large to be in Hardy-Weinberg equilibrium. Although drift occurs in all populations, the smaller the population, the greater the change is in allele frequencies from generation to generation as a result of genetic drift. If the gametes of only a few individuals form the next generation of alleles, the alleles they carry may by chance not be representative of the parent population from which they were drawn, as illustrated in figure 20.5. In this example, the small number of individuals are removed from a bottle. By chance, most of the individuals removed are green. So the new population has a much higher population of green individuals than the parent generation had. Suppose, for example, that a population in which dominant B and recessive B are equally represented. 4 individuals formed in the next generation, and that by chance they were 2 heterozygotes and 2 homozygotes that are homozygous dominant. That is, the allele frequencies in the next generation would be for P 0.75 and for Q 0.25. In fact, if you were to replicate this experiment 1,000 times, each time randomly drawing 4 individuals from the parental population, then in about 8 of the 1,000 experiments, one of the 2 alleles would be missing entirely. This result leads to an important conclusion. Genetic drift can often lead to loss of alleles in isolated populations. Alleles that initially are uncommon are particularly vulnerable. A set of small populations that are isolated from one another may come to differ strongly as a result of genetic drift, even if the forces of natural selection are the same for all. Because of genetic drift, sometimes harmful alleles may increase in frequency in small populations, despite the selective disadvantage, and favorable alleles may be lost even though they are selectively advantageous. Although genetic drift occurs in any population, its effect is inversely related to population size. Thus, it is particularly likely in populations that were founded by a few individuals, or in which the population was reduced to a very small number at some time in the past. Such founder effects or bottlenecks are particularly clear examples of genetic drift. Sometimes one or a few individuals disperse and become the founders of a new isolated population at some distance from their place of origin. These pioneers are not likely to carry all of the alleles present in the source population. Thus, some alleles may be lost from the new population, and others may change drastically in frequency. In some cases, previously rare alleles in the source population may become a significant fraction of the new population's genetic endowment. This phenomenon, in which the allele frequencies of the founding individuals are different from the allele frequencies of the population from which they came, is called the founder effect. Founder effects are not rare in nature. Many self-pollinating plants start new populations from a single seed. Founder effects have been particularly important in the evolution of organisms on distant oceanic islands, such as the Hawaiian and Galapagos Islands. Most of the organisms in such areas probably derive from one or a few initial founders. Although rare, such events are occasionally observed, such as when the mass of vegetation carrying several iguanas washed up on the shore of the Caribbean island of Anguilla in 1996, leading to the establishment of a population that still occurs there to this day. In a similar way, isolated human populations begun by relatively few individuals are often dominated by genetic features characteristic of their founders. Amish populations in the United States, for example, were established from relatively few individuals and have unusually high frequencies of a number of conditions, such as polydactylism, the presence of a 6th finger. Even if organisms do not move from place to place, occasionally their populations may be drastically reduced in size. This may result from flooding, drought, epidemic disease, and other natural forces, or from changes in the environment. Just as we have seen with the founder effect, the few surviving individuals may by chance have allele frequencies different from those in the entire population, and some alleles may be lost entirely. The resulting alterations and loss of genetic variability have been termed the bottleneck effect. The genetic variation of some living species appear to be severely depleted, probably as a result of a bottleneck effect in the past. For example, the northern elephant seal, which breeds on the western coast of North America and nearby islands, was nearly hunted to extinction in the 19th century and was reduced to a single population containing perhaps no more than 20 individuals on the island of Guadalupe off the coast of Baja California. The island of Guadalupe off the coast of Baja California. Guadalupe? Excuse me for the mispronunciation. As a result of this bottleneck, the species has lost almost all of its genetic variation, even though the seal populations have rebounded and now number in the tens of thousands and breeding locations as far north as San Francisco. Anytime a population becomes drastically reduced in numbers, such as an endangered species, the bottleneck effect is a potential problem. Even if the population size rebounds, the lack of variability means that the species remains vulnerable to extinction. Selection. As Darwin pointed out, some individuals leave behind more progeny than others, and the rate at which they do so is affected by their phenotype. We describe the results of this process as selection, when individuals with one phenotype leave, on average, more surviving offspring in the next generation than individuals with an alternative phenotype. Please see figure 20.4e. In artificial selection, a breeder selects for the desired traits or characteristics. In natural selection, the environmental conditions determine which individuals in a population produce the most offspring. Evolution by natural selection occurs when the following conditions are met. Phenotypic variation must exist among individuals in a population. So natural selection works by favoring individuals with some traits over individuals with alternative traits. If no variation exists, natural selection cannot operate. Variation among individuals must result in differences in the number of offspring surviving in the next generation. This is natural selection. Because of their phenotype or behavior, some individuals are more successful than others in producing offspring. Although many traits are phenotypically variable, individuals exhibiting variation do not always differ in survival and reproductive success. Phenotypic variation must have a genetic basis. For natural selection, please see item 2, to result in evolutionary change. The selected differences must have a genetic basis. Not all variation has a genetic basis. Even genetically identical individuals may be phenotypically quite distinctive if they grew up in different environments. Such environmental effects are common in nature. In many turtles, for example, individuals hatched from eggs laid in moist soil are heavier and longer with wider shells than individuals from nests in drier areas. When phenotypically different individuals do not differ genetically, differences in the number of offspring will not alter the genetic composition of the population in the next generation, and thus no evolutionary change will have occurred. It is important to remember that natural selection and evolution are not the same. It is important to remember that natural selection and evolution are not the same. The two concepts are often incorrectly equated. Natural selection is a process, whereas evolution is the historical record or the outcome of change through time. So, natural selection is what allows evolution to occur, or it's evolution by means of natural selection, if you can get it that way. So natural selection can lead to evolution because natural selection is the process and evolution is the outcome. But natural selection is only one of the several processes that can result in evolutionary change. Moreover, natural selection can occur without producing evolutionary change. Only if variation is genetically based will natural selection lead to evolution. The result of evolution is driven by natural selection is that populations become better adapted to their environment. Many of the most dramatic documented instances of adaptation involve genetic changes that decrease the probability of capture by a predator. Caterpillar larvae of the common sulfur butterfly usually exhibit a pale green color, providing excellent camouflage against the alfalfa plants on which they feed. An alternative bright yellow color morph is reduced to a very low frequency because this color renders the larva highly visible on the food plant, making it easier for bird predators to see them. Please see figure 20.7 and even figure 20.4e. One of the most dramatic examples of background matching involves ancient lava flows in the desert of the American Southwest. Reminds me of the trip I had to Albuquerque, New Mexico. In these areas, the black rock formations produced when the lava cooled contrast startled with the surrounding bright glare of the desert sand. In class, you can see this if you were to go to New Mexico, to Albuquerque, and go north from Albuquerque to Santa Fe. Populations of many species of animals occurring on these rocks, including lizards, rodents, and a variety of insects, are dark in color, whereas sand-dwelling populations in surrounding areas are much lighter. Please see figure 20.7. Predation is the likely cause for these differences in color. Laboratory studies have confirmed that predatory birds, such as owls, are adept at picking out individuals occurring on backgrounds to which they are not adapted, such as the mice you see there. They are referred to as being the Snickers bar of the desert, being eaten by owls especially. Many studies of selection have focused on genes encoding enzymes because in such cases the investigator can directly assess the consequences to the organism of changes in the frequency of alternative enzyme alleles. Often investigators find that enzyme allele frequencies vary with latitude, so that one allele is more common in the northern populations but is progressively less common at more southern locations. A superb example is seen in the studies of a fish, the mummic cog from the genus Fundulus, which ranges along the eastern coast of North America. In this fish, geographic variation occurs in allele frequencies for the gene that produces the enzyme lactate dehydrogenase, which catalyzes the conversion of pyruvate into lactate. Biochemical studies show that the enzymes formed by these alleles function differently at different temperatures, thus explaining their geographic distributions. The form of the enzyme more frequent in the north is a better catalyst at low temperatures than is the enzyme from the south. Moreover, studies indicate that at low temperatures, individuals with the northern allele swim faster and presumably survive better than individuals with the alternative allele. A particularly clear example of selection in natural populations is provided by studies of pesticide resistance in insects. The widespread use of insecticides has led to the rapid evolution of resistance in more than 500 pest species. The cost of this evolution in terms of crop losses and increased pesticide use has been estimated at $3 to $8 billion per year. In the housefly, the resistance allele of the PIN gene decreases uptake of the insecticide, whereas alleles of the KDR and the DLDR genes decrease the number of target sites, thus decreasing the binding ability of the insecticide, which you all can see in figure 20.8 shown here. Other alleles enhance the ability of the insect's enzymes to identify and detoxify insecticide molecules. Single genes are also responsible for resistance in other organisms. For example, nori rats are normally susceptible to the pesticide warfarin, which diminishes the clotting ability in the rat's blood, leading to fatal hemorrhaging. However, the resistance allele of the VKORC1 gene reduces the ability of warfarin to bind to its target enzyme and thus renders it ineffective. Selection imposed by humans has also led to the evolution of resistance to antibiotics in many disease-causing pathogens. For example, Staphylococcus aureus, which causes staph infections, was initially treated by penicillin, which latched onto Staphylococcus aureus, degrading cell walls and causing death of the bacteria. However, within 4 years of mass production of the drug, evolutionary change in Staphylococcus aureus modified an enzyme penicillinase so that it would attack penicillin, making the drug unable to attach to Staphylococcus aureus and thus rendering it ineffective. Since that time, several other drugs have been developed to attack the microbe, and each time resistance has evolved. As a result, staph infections have reemerged as a major health threat. The speed by which the adaptation occurs may be surprising, but remember that microbes have enormous population sizes. Even though mutation rates are low, the vast size of these populations guarantees a steady supply of new mutations for natural selection to utilize. The effect of such evolution on human health is enormous. In the United States alone, 2 million people die each year. 2 million people in the United States alone, 2 million people each year become ill due to antibiotic-resistant bacteria, and 23 million die from such infections. So 2 million people are ill yearly due to antibiotic-resistant bacteria, and 23,000 die from it each year. Selection occurs when individuals with one phenotype leave more surviving offspring in the next generation than individuals with an alternative phenotype. Evolutionary biologists quantify reproductive success as fitness, the number of surviving offspring left in the next generation. Fitness is a relative concept. The most fit phenotype is simply the one that produces on average the greatest number of offspring. Suppose, for example, in a population of toads, 2 phenotypes exist, green and brown. Suppose further that green toads leave on average 4 offspring in the next generation, but brown toads leave only 2.5. By custom, the most fit phenotype is assigned a fitness value of 1.0, and other phenotypes are expressed as relative proportions. In this case, the fitness of the green phenotype would be 4.0 divided by 4.0, giving us 1.0000. And the fitness of the brown phenotype would be 2.5 divided by 4, which is 0.625, as you see there. The difference in fitness would therefore be 1 minus 0.625, which is 0.375. A difference in fitness of 0.375 is quite large. Natural selection in this case strongly favors the green phenotype. If differences in color have a genetic basis, then we would expect evolutionary change to occur. The frequency of green toads should be substantially greater in the next generation. Further, if the fitness of the 2 phenotypes remained unchanged, we would expect alleles for the brown phenotype eventually to disappear from the population. Fitness may consist of many components, although selection is often characterized as survival of the fittest. Differences in survival are only one component of fitness. Even if no differences in survival occur, selection may operate if some individuals are more successful than others in attracting mates. In many territorial animal species, for example, large males mate with many females, and small males rarely get to mate. Selection with respect to mating success is termed sexual selection. In addition, the number of offspring produced per reproductive event is also important. Large female frogs and fish lay more eggs than do smaller females, and thus they may leave more offspring in the next generation. Fitness is therefore a combination of survival, mating success, and number of offspring per mating. Selection favors phenotypes with the greatest fitness, but predicting fitness from a single component can be tricky because traits favored for one component of fitness may be at a disadvantage for others. As an example, in water striders, larger females lay more eggs per day. Thus, natural selection at this stage favors large size. However, larger females also die at a younger age and thus have fewer opportunities to reproduce than smaller females. Overall, the two opposing directions of selection cancel each other out, and the intermediate-sized females leave the most offspring in the next generation. So you have here the data. So length of female water strider in millimeter, shown there in green, and number of eggs laid per day. The very same shown here. Length of adult female water strider in millimeter. However, in yellow, it's lifespan of adult female in days. And then lastly, still we have length of adult female water strider in millimeters. And we have number of eggs laid during lifetime. Males and females have the common goal of improving the quantity and quality of offspring they produce, but usually differ in the way they attempt to maximize fitness. Such a difference in reproductive behavior is clearly seen in mate choice. Darwin was the first to observe that females often do not mate with the first male they encounter, but instead seem to evaluate a male's quality and then decide whether to mate. Peons prefer to mate with peacocks that have more eye spots on their elaborate tail feathers. Similarly, female frogs prefer to mate with males having more acoustically complex and thus attractive calls. This behavior, called mate choice, is well known in many invertebrate and vertebrate species. So you see here products of sexual selection. The peacock at A, and then the Rihanna bird of paradise in B. And then at C, we have female peons preferring to mate with males having greater numbers of eye spots on their tails. Males are selective when choosing a mate less frequently than females. Why should this be? Many of the differences in reproductive strategies between sexes can be understood by comparing the parental investment made by males and females. Parental investment refers to the energy and time each sex invests in producing and rearing offspring. It is, in effect, an estimate of the energy expended by males and females in each reproductive event. Numerous studies have shown that females generally have a higher parental investment. One reason is that the eggs are much larger than sperm, 195,000 times larger in humans. Eggs contain protein and lipids in the yolk and other nutrients for the developing embryo, but sperm are little more than mobile DNA packages. In some groups of animals, mammals for example, females are responsible for gestation and lactation, costly reproductive functions only they can carry out. The consequence of such inequalities in reproductive investment is that the sexes face very different selection pressures. Because any single reproductive event is relatively inexpensive for males, they can best increase their fitness by mating with as many females as possible. This is because male fitness is likely not limited by the amount of sperm they can produce. By contrast, each reproductive event for females is much more costly, and the number of eggs that can be produced often limits reproductive success. For this reason, a female should be choosy trying to pick a male that can provide the greatest benefit to her offspring and thus improve her fitness. These conclusions hold only when female reproductive investment is much greater than that of males. In species with biparental care, males may contribute equally the cost of raising young. In this case, the degree of mate choice should be more equal in the sexes. In some cases, male investment exceeds that of females. For example, in the male moron crickets transfer a protein-containing packet a spermatophore, to females during mating. Almost 30% of a male's body weight is made up by the spermatophore, which provides nutrition for the female and helps her develop her eggs. As you might expect from our model of mate choice, in this case, it is the females that compete with one another for access to males, which are the choosy sex. Indeed, males are quite selective, favoring heavier females. Heavier females have more eggs, thus males choose larger females, leave more offspring. Males care for eggs and developing young in many species, including seahorses and a number of birds and insects, as with Mormon crickets. These males are often choosy, and females compete for mates. There you have it. The reproductive success of an individual is determined by how long the individual lives, how frequently it mates, and how many offspring it produces per mating. The second of these factors, competition for mates, is termed sexual selection. So as female body weight increases, it looks to be an increase in the number of mature eggs. Some people consider sexual selection to be distinctive from natural selection, but others see it as a subset of natural selection, just one of the many factors affecting an organism's fitness. Sexual selection involves both intrasexual selection, or competitive interactions between members of one sex, and intersexual selection, which is another name for mate choice. Sexual selection leads to the evolution of structures used in combat with males, such as deer's antlers and ram's horns, as well as ornaments used to persuade members of the opposite sex to mate, such as long tail feathers and bright plumage. These traits are called secondary sexual characteristics. Selection strongly favors any trait that confers greater ability in mate competition. Larger body size is a great advantage if dominance is important, as is in territorial spaces. Males may thus be considerably larger than females. Such differences between the sexes are referred to as sexual dimorphism. In other species, structures used for fighting, such as horns, antlers, and large canine teeth, have evolved to be larger in males because of the advantage they give in intrasexual competition. Sometimes sperm competition occurs between sperm of different males if females mate with multiple males. This type of competition, which occurs after mating, has selected for features that increase the probability that a male sperm will fertilize the eggs. Such features include sperm transfer organs designed to remove the sperm prior to mating, large testes to produce more sperm per mating, and sperm that hook themselves together to swim more rapidly. In many species, individuals of one sex, usually males, compete with one another for the opportunity to mate. Competition among males can occur for territory in which females feed or bear young. Males may also directly compete for the females themselves. A few successful males may engage in an inordinate number of matings, whereas most males do not mate at all. For example, elephant seal males control territories on breeding beaches, and a few dominant males do most of the breeding. On one beach, for example, 8 males impregnated 348 females, whereas the remaining males mated rarely, if at all. Intersexual selection concerns the active choice of a mate. Mate choice has both direct and indirect benefits. In some cases, the female benefits directly from her choice of mates. If mates help raise offspring, females benefit by choosing the mate that can provide the best care. The better the parent, the more offspring she is likely to rear. In other species, males provide no care, but maintain territories that provide food, nesting sites, and predator refuges. In red deer, males that hold territories with highest quality grasses mate with the most females. In this case, there is a direct benefit of a female mating with a territory owner. She feeds with little disturbance on quality food. In many species, however, males provide no direct benefits of any kind to females. In such cases, it is not intuitively obvious what females have to gain by being choosy. Moreover, what could be the possible benefit of choosing a mate with an extremely long tail or a complex song? One possible indirect benefit of mate choice is that it could lead to higher quality offspring. A number of theories have been proposed as to how this might work. One idea is that females choose the male that is the healthiest or oldest. Large males, for example, have probably been successful at living long, acquiring a lot of food, and resisting parasites and disease. In other species, features other than size may indicate a male's condition. In guppies and birds, the brightness of a male's color reflects the quality of his diet and overall health. Females may gain two benefits from mating with the healthiest males. First, healthy males are less likely to carry disease, which might be transmitted to the female during mating. This would be a direct benefit. Second, to extend the male's success in living long and prospering is a result of his genetic makeup. The female will be ensuring that her offspring receive good genes from their father, an indirect benefit. Several experimental studies in fish and moths have examined whether female mate choice leads to greater reproductive success. In these experiments, females in one group were allowed to choose males with which they would mate, whereas males were randomly mated to a different group of females. Offspring of females that chose their mates were more vigorous and survived better than offspring from females given no choice, which suggests that females preferred males with a better genetic makeup. A variant of this theory goes one step further in some cases. Females prefer mates with traits that appear to be detrimental to survival. The long tail of the peacock is a hindrance in flying and makes males more vulnerable to predators. Why should females prefer males with such traits? The handicap hypothesis states that only genetically superior mates can survive with such a handicap. By choosing a male with the largest handicap, the female is ensuring that her offspring will receive these quality genes. Of course, the male offspring will also inherit the genes for the handicap. For this reason, evolutionary biologists are still debating the merit of this hypothesis. Alternative theories about evolution of mate choice include some courtship displays appear to have evolved from a predisposition in the female's sensory system to respond to certain stimuli. For example, females may be better able to detect particular colors or sounds at a certain frequency and thus be attracted to such signals. Sensory exploitation involves the evolution in males of a signal that exploits these pre-existing biases. For example, if females are particularly adept at detecting red objects, then red coloration may evolve in males as part of a courtship display. To understand the evolution of courtship calls, consider the vocalization of the Tungara frog. Unlike related species, males include a short burst of sound, termed chuck, at the end of the calls. Recent research suggests that not only are females of this species particularly attracted to calls of this sort, but so are females of related species, even though males of these species do not produce chucks. This pattern suggests that the preference evolved for some reason in the ancestor of all of the species, and then subsequently led to the evolution of a chuck only in males of the Tungara frog. Why males of other species haven't evolved to produce the chuck? It's a good question. The opportunity for sensory exploitation may be widespread. For example, researchers conducted an experiment to see if birds had latent preferences for a particular stimuli using a common bird in the pet trade, the zebra finch. To conduct the experiment, the researchers glued long feathers vertical to the tops of the heads of male birds. The result was that the males had very tall crests, completely unlike anything seen in nature. They then presented males with different colored crests to females to see if the females had preference for one color over another. Surprisingly, females were strongly attracted to males with white crests but not green or red ones. This might be why it's not at all clear. It suggests that there is something in the zebrafinch's neural hardwiring that for some reason finds white much more attractive than green or red, regardless of the cause of the preference. This finding suggests that if a mutation arose that caused a male to have a white crest on his head, the mutation would stow great advantage that would quickly spread through the population. Perhaps there are many such latent preferences in species, and then preferences we might never be able to predict that might be hard to explain. But if appropriate variation arises in a population, females may prefer it. This is a fascinating area of research that is just in its infancy and will require coupling studies of neurophysiology and behavior to understand how and why such mate choice preferences evolve. So a great variety of other hypotheses have been proposed to explain the evolution of mating preferences. Many of these hypotheses may be correct in some circumstances, but none seems capable of explaining all the variation in mating behavior in the animal world. This is an area of vibrant research with new discoveries appearing regularly. Natural selection has been discussed as a process that removes variation from a population by favoring one allele over others at a gene locus. However, in some circumstances, selection can do exactly the opposite and actually maintain population variation. In some circumstances, the fitness of a phenotype depends on its frequency within the population, a phenomenon termed as frequency-dependent selection. This type of selection favors certain phenotypes depending on how commonly or uncommonly they occur. In negative frequency-dependent selection, rare phenotypes are favored by selection. Thus, fitness has a negative relationship with phenotype frequency, hence the term negative frequency-dependent. Assuming the genetic basis for the phenotypic variation, such selection will have the effect of making rare alleles more common, thus maintaining variation. Negative frequency-dependent selection can occur for many reasons. For example, it is well known that animals or people searching for some form of search image, that is, they become particularly adept at picking out certain objects. Consequently, predators may form a search image for common prey phenotypes. Rare forms may thus be preyed upon less frequently. An example is fish predation on an insect. The water boatman, which occurs in 3 different colors, experiments indicate that each color types is preyed upon disproportionately when it is the most common one. Fish eat more of the common colored insects than would occur by chance alone. Another cause of negative frequency dependence is resource competition. If genotypes differ in their resource requirements, as occurs in many plants, then the rarer genotype will have fewer competitors. When the different resource types are equally abundant, the rarer genotype will be at an advantage relative to the more common genotype. Positive frequency-dependent selection has the opposite effect. By favoring common forms, it tends to eliminate variation from a population. For example, predators don't always select common individuals. In some cases, oddballs stand out from the rest and attract attention. The strength of selection should change through time as a result of frequency-dependent selection. In negative frequency-dependent selection, rare genotypes should become increasingly common, and their selection advantage should decrease correspondingly. Conversely, in positive frequency dependence, the rarer the genotype becomes, the greater the chance it will be selected against. In some cases, selection favors one phenotype at one time and another phenotype at another time, a phenomenon called oscillating selection. If selection repeatedly oscillates in this fashion, the effect will maintain genetic variation in the population. One example concerns the medium groundfinch of the Galapagos Islands. In times of drought, the supply of small soft seeds is depleted, but there are still enough large seeds around. Consequently, birds with big bills are favored. However, when wet conditions return, the ensuing abundance of small seeds favors birds with smaller bills. Oscillating selection and frequency-dependent selection are similar because, in both cases, the form of selection changes through time. But it is important to recognize that they are not the same. In oscillating selection, the fitness of a phenotype does not depend on its frequency. Rather, environmental changes lead to the oscillation in selection. In contrast, in frequency-dependent selection, it is the change in frequencies themselves that lead to changes in fitness of different phenotypes. If heterozygotes are favored over homozygotes, then natural selection actually tends to maintain variation in the population. This heterozygote advantage favors individuals with copies of both alleles and thus works to maintain both alleles in the population. Some evolutionary biologists think the heterozygote advantage is pervasive and can explain the high levels of genetic variation observed in natural populations. Others, however, consider it to be relatively rare. The best documented example of the heterozygote advantage is sickle cell anemia, a hereditary disease affecting hemoglobin in humans. Individuals with sickle cell anemia exhibit symptoms of severe anemia and abnormal red blood cells that are irregular in shape, with a great number of long sickle-shaped cells. The average incidence of the dominant S allele in Central African populations is about 0.12, far higher than that found among Americans of African descent. From the Hardy-Weinberg principle, you can calculate that 1 in 5 Central African individuals is heterozygous at the dominant S allele, and 1 in 100 is homozygous and develops the fatal form of the disorder. People who are homozygous for the sickle cell allele almost never reproduce because they usually die before they reach reproductive age. Why then is the dominant S allele not eliminated from the Central African population by selection rather than being maintained at such high levels? As it turns out, one of the leading causes of illness and death in Central Africa, especially among young children, is malaria. People who are heterozygous for the sickle cell allele and thus do not suffer from sickle cell anemia are much less susceptible to malaria. The reason is that when the parasite that causes malaria, Plasmodium falciparum, enters a red blood cell, it causes extremely low oxygen tension in the cell, which leads to the sickling in the cells of individuals either homozygous or heterozygous for a sickle cell allele, but not individuals that do not have the sickle cell allele. Such cells are quickly filtered out of the bloodstream by the spleen, thus eliminating the parasite. Thus, homozygotes with the sickle cell allele all die of anemia. Homozygotes with the non-sickle cell allele are vulnerable to malaria, and heterozygotes have the highest fitness. Consequently, even though most homozygous recessive individuals die at a young age, the sickle cell allele is maintained at high levels in these populations because it is associated with resistance to malaria in heterozygotes, and also, for reasons not yet fully understood, with increased fertility in female heterozygotes. Figure 20.17 shows the overlap between regions where sickle cell anemia is found and those where malaria is prevalent. For people living in areas where malaria is common, having the sickle cell allele in the heterozygous condition has adaptive value. However, in the United States, the environment does not place a premium on the resistance to malaria because the disease is now essentially absent from North America. Consequently, no adaptive value counterbalances the ill effects of the disease. In this non-malarial environment, selection is acting to eliminate the dominant S allele. Only 1 in 375 Americans of African descent, many of whose ancestors lived in Central Africa, develops sickle cell anemia, far fewer than in Central Africa. In nature, many traits, perhaps most, are affected by more than one gene. The interactions between genes are typically complex, as you saw in prior chapters. For example, alleles of hundreds of genes play a role in determining human height. And that's hundreds of genes. In such cases, selection operates on all genes, influencing most strongly those who make the greatest contribution to the phenotype. Such traits exhibit a continuous distribution of phenotypes, often conforming to a normal curve rather than several discretely different. In some situations, selection acts to eliminate intermediate types, a phenomenon called disruptive selection. See Figure 20.18a. A clear example is the different beak sizes of the African black-bellied seedcracker finch. Populations of these birds contain individuals with large and small beaks, but very few individuals with intermediate-sized beaks. As the name implies, these birds feed on seeds, and the available seeds fall into two size categories, large and small. Only large-beaked birds can open the tough shells of large seeds, whereas birds with smaller beaks are more adept at handling small seeds. Birds with intermediate-sized beaks are at a disadvantage with both types. They are unable to open the large seeds and too clumsy to efficiently process the small seeds. Consequently, selection acts to eliminate the intermediate phenotypes, in effect partitioning or disrupting the population into two phenotypically distinct groups. Directional selection. Please see Figure 20.18b. When selection acts to eliminate one extreme from an array of phenotypes, the genes promoting this extreme becomes less frequent in the population and may eventually disappear. This form of selection is called directional selection. Thus, in the Drosophila population illustrated in Figure 20.20, eliminating flies that move toward light causes the population over time to contain fewer individuals with alleles promoting such behavior. If you were to pick an individual at random from a later generation of flies, there is a smaller chance that the fly would spontaneously move toward light than if you had selected a fly from the original population. Artificial selection has changed the population in the direction of being less attracted to light. Directional selection often occurs in nature when the environment changes, favoring traits at one end of the phenotypic distribution. Lastly, stabilizing selection, shown at Figure 20.18c. When selection acts to eliminate both extremes from an array of phenotypes, the result is to increase the frequency of the already common intermediate type. This form of selection is called stabilizing selection. In effect, the selection is operating to prevent change away from the middle ranges of values. Selection does not change the most common phenotype of the population, but rather makes it even more common by eliminating the extremes. Many examples are known. In humans, infants with intermediate birth weight have the highest survival rate. In ducks and chickens, eggs of intermediate weight have the highest hatching success. So there are experimental studies on natural selection that I will not go over at the moment. However, I will get on into interactions among evolutionary forces. The amount of genetic variation in a population may be determined by the relative strength of different evolutionary processes. Sometimes these processes act together in other way, and in other cases they may work in opposition. In theory, if dominant allele B mutated to recessive allele B at a high enough rate, allele B that's recessive could be maintained in the population, even if natural selection strongly favored the dominant allele B. In nature, however, mutation rates are rarely high enough to counter the effects of natural selection. The effect of natural selection also may be countered by genetic drift. Both of these processes may act to remove variation from a population. But selection is a non-random process that operates to increase the representation of alleles that enhance survival and reproductive success, whereas genetic drift is a random process in which any allele may increase. Thus, in some cases, drift may lead to a decrease in the frequency of the allele that is favored by selection. In some extreme cases, drift may even lead to the loss of a favorite allele from a population. Remember, however, that the magnitude of drift is inversely related to the population size. Consequently, natural selection is expected to overwhelm drift, except when populations are very small. Gene flow can either be a constructive or constraining force. On one hand, gene flow can spread a beneficial mutation that arises in one population to others. On the other hand, gene flow can impede adaptation within one population by continual flow of inferior alleles. Consider two populations of a species that live in different environments. In this situation, natural selection may favor different alleles, a dominant bee and a recessive bee, in two populations. In the absence of other evolutionary processes such as gene flow, the frequency of the dominant bee would be expected to reach 100% in one population and 0% in the other. However, if gene flow occurred between the two populations, then the less favored allele will continually be reintroduced into each population. As a result, the frequency of the alleles in the populations would reflect the balance between the rate at which gene flow brings the inferior allele into the population and the rate at which natural selection removes it. A classic example of gene flow opposing natural selection occurs on abandoned mine sites in Great Britain. Although mining activities ceased hundreds of years ago, the concentration of metal ions in the soil is still much greater than the surrounding areas. Large concentrations of heavy metals are generally toxic to plants, but alleles of certain genes confer ability to grow on soils in heavy metals. The ability to tolerate heavy metals comes at a price, however. Individuals with the resistance allele exhibit lower growth rates on non-polluted soils. Consequently, we would expect the resistance allele to occur with frequency of 100% on the former sites and 0% elsewhere. Heavy metal tolerance has been studied intensively. In the slender bent grass, in which resistance allele occurs at the intermediate levels in many areas. The explanation relates to the reproductive system of the grass, in which pollen, the flow equivalent of sperm, is dispersed by the wind. As a result, pollen grains and the alleles they carry can move great distances, leading to novel levels of gene flow between mine sites and unpolluted areas, high enough to counteract the effects of natural selection. In general, the extent to which gene flow can hinder the effects of natural selection should depend on the relative strength of two processes. In species in which gene flow is generally strong, such as birds and wind-pollinated plants, the frequency of the allele less favored by natural selection may be relatively high. In more sedentary species that exhibit low levels of gene flow, such as snails, the favored allele frequency should occur at a frequency near 100%. Although selection is the most powerful of the principal agents of genetic change, there are limits to what it can accomplish. These limits result from multiple phenotypic effects of alleles, lack of genetic variation upon which selection can act, and interactions between genes. Alleles often affect multiple aspects of phenotype. Think back to pleiotropy. These multiple effects tend to set limits on how much a phenotype can be altered. For example, in chickens, the same gene affects the size of a hen's comb and the rate at which she lays. Alleles that produce large combs also leads to faster egg production. As a result of this linkage, selection for hens that lay many eggs but may have small combs would be very difficult. Genetic variation is a prerequisite for evolutionary change. Without it, natural selection cannot produce evolution. Over 80% of the gene pool of thoroughbred horses racing today goes back to 31 ancestors from the late 18th century. Despite intense directional selection on thoroughbreds, their performance times have not improved for more than 60 years. Decades of intense selection presumably have removed variation from the population at a greater rate than mutation can replenish it, such that little genetic variation now remains and evolutionary change is not possible. In some cases, phenotypic variation for a trait may never have had a genetic basis. The compound eyes of insects are made up of hundreds of visual tints termed ommatidia. In some individuals, the left eye contains more ommatidia than the right. In other individuals, the right contains more than the left. However, despite the intense selection experiments in the laboratory, scientists have never been able to produce a line of fruit flies that consistently has more ommatidia in the left than the right eye. The reason is that the separate genes do not exist for the left and right eyes, but the same genes affect both eyes, and the differences in the number of ommatidia results from differences that occur as the eyes are formed in the development process. Thus, despite the existence of phenotypic variation, no underlying genetic variation is available for selection to favor. And as discussed in Chapter 12, epistasis is the phenomenon in which an allele for one gene may have different effects depending on alleles present in other genes. Because of epistasis, the selective advantage of an allele at one gene may vary from one genotype to another. If a population is polymorphic for a second gene, then selection for the first gene may be constrained because the different alleles are favored in different individuals of the same populations. Studies on bacteria illustrate how selection on alleles for one gene depend on which alleles are present at other genes. In E. coli, two biochemical pathways exist to break down gluconate, each using enzymes produced by different genes. One gene produces the enzyme 6-PGD, for which there are several alleles, and then when common allele for the second gene, which codes for the other biochemical pathway, is present, selection does not favor one allele over the other at the 6-PGD. In some E. coli, however, an alternative allele at the second gene occurs that is not functional. The bacteria with this alternative allele are forced to rely on the 6-PGD pathway, and in this case, selection favors the 6-PGD allele over another. Thus, epistatic interactions exist between the two genes, and the outcome of the natural selection on the PGD gene depends on which alleles are present at the second gene. So this has been your instructor, Skylar Huff. Thank you all for listening. Take notes. Make sure you all understand how changes in allele frequency occur, those 5 agents of evolutionary change, including those reproductive strategies. So this